So, structure of DNA. This is B type DNA and this is double stranded DNA. So, the DNA term DNA given by Jackaris, given by the scientist Jackaris. Next, the DNA structure is given by two scientists. The structure of DNA is given by two scientists, Watson and Crick. That's why they got Nobel Prize. So DNA is a polynucleotide chain. Many nucleotides are joined together to formation of a DNA structure. So in case of one nucleotide, we have observed this is one nucleotide, means this polynucleotide chain is mainly made up of three components. One is pentose sugar, nitrogen base, and as well as third one phosphoric acid. These three things to make up one nucleotide. So these three things are to make up single nucleotide. So these single nucleotides are many or joined together to formation of a DNA structure. Consist of 5 carbons, carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5 is as a R. This carbon number 5 consists of CH2OH as a freely attached group. And a third carbon mainly consists of, that is OH as a functional group. This is OH as a functional group, and next nucleotide is attached with this place, that is third carbon. Next, this CH2OH is connected with phosphoric acid. Phosphoric acid. This phosphoric acid mainly consists of oxygen and hydroxyl group. 3 hydroxyl groups are present. So nitrogen base which is attached with this particular place, the first carbon is attached with that is nitrogen base. Nitrogen base which is attached with first carbon. So nitrogen base attached to first carbon of pentose sugar. Next, phosphoric acid or phosphate group is attached with fifth carbon. So next, phosphate group or phosphoric acid attaches to at fifth carbon of pentose sugar. In this case, pentose sugar is deoxyribose sugar. So this is deoxyribose sugar. Next, the bond is formed between carbon number 1 and nitrogen base. This is glycosidic bond which is formed between the first carbon of pentose sugar and nitrogen base, either adenine, guanine, thymine, cytosine. These nitrogen bases are two types of nitrogen bases are attached. One is purines are attached as well as pyrimidines. Purines and pyrimidines are attached. In case of purines, 9th position of purine attaches with the first carbon of pentose sugar. In case of pyrimidines, the nitrogen atom, first position of nitrogen atom connected with the first carbon of pentose sugar. Here purines means they are mainly 9th position of purines connected with the first carbon of pentose sugar. In case of pyrimidines, first position of pyrimidines connected with the first carbon of pentose sugar. Next, phosphoric acid connected with the pentose sugar by the fifth carbon. This is fifth carbon. Here fifth carbon, pentose sugar of hydrogen attaches with the hydroxyl group of pentose sugar. Means phosphoric acid of hydrogen attaches with pentose sugar of hydroxyl group at fifth carbon is attached and releases water molecule. This bond is called, that is, phosphodiester bond. Phosphodiester bond is formed because hydrogen of phosphoric acid and hydroxyl group of pentose sugar both are joined together, form of a phosphodiester bond is formed and releases water molecule. That bond is phosphodiester bond is present. This is about one nucleotide, and many nucleotides we have to discuss in, and we have to draw one by one. Mainly polynucleotides, this is about one nucleotide, and we have to draw different nucleotides, many nucleotides. So this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. Here CH2OH is freely attached. Here first carbon attaches with adenine, means first carbon attaches with first nitrogen base, that is adenine. Before that, purines and pyrimidines are 1:1 ratio. Purines and pyrimidines are 1:1 ratio. Purines and pyrimidines. So in case of adenine, guanine is called purines. Next, pyrimidines means cytosine, thymine. These are pyrimidines. 1:1 ratio. Adenine binds with thymine. Adenine binds with thymine by the double hydrogen bond. Next, cytosine binds with guanine by the triple hydrogen bond. Nitrogen bases are attaches by the hydrogen bonds. Adenine nitrogen base attaches with thymine by the help of double nitrogen bond. Next, cytosine attaches with guanine by the triple hydrogen bond. This is one thing. So here adenine binds with thymine and cytosine binds with guanine by the double bond and triple bond of hydrogen bonds. This is one. Here third carbon consists of functional group, and next nucleotide attaches with third carbon because of functional group is present. Next, this is next nucleotide. Carbon number 1 attaches with nitrogen base. That nitrogen base is nothing but cytosine. Next, third one, this contain functional group, and fifth one, that is CH2OH is freely attaches with fifth carbon, and here this OH is connected with phosphoric acid. Phosphoric acid connected with this OH. Phosphoric acid connected with this OH and releases water molecule by the process of phosphodiester bond formation. Next, one more nucleotide, that is here carbon number 1 attaches with adenine, thymine, cytosine, guanine, and thymine attaches with thymine. Next, third carbon mainly functional group, and fifth carbon, that is CH2OH is freely attached. Next, before nucleotide connected with that is phosphate group. Phosphate group are connected with CH2OH of fifth carbon by the phosphodiester bond. Here one bond is present, nitrogen base and pentose sugar, that is glycosidic bond. Here glycosidic bond, here glycosidic bond, and one more one. This is another nucleotide. This is carbon number 1 attaches with guanine and by the glycosidic bond. Next, carbon number 3 consists of OH is a functional group, and carbon number 5 attaches with CH2OH is freely attached. Next, this OH is connected with that is phosphoric acid. This phosphoric acid connected with this CH2OH of fifth carbon by the help of phosphodiester bond, releases water molecule. About this is one helix. This is about one helix. This is 5th prime this is third prime, this is fifth prime. Two primes are present in DNA structure, one is third prime and one more is fifth prime. Next, another helix. It is anti-parallel direction. So it is anti-parallel direction. This is carbon number 1, carbon number 2, carbon number 3, carbon number 4 and carbon number 5. Carbon number 1 connected with that is nitrogen base. Adenine always binds with thymine. So double bond thymine. This is thymine. First carbon attaches with thymine. Third carbon contain that is OH as a functional group, and fifth carbon consists of CH2OH is a freely attached at fifth carbon. Here glycosidic bond is formed. Next, another nucleotide. So this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. Carbon number 1 attaches with another opposite nucleotide, that is cytosine binds with guanine, triple bond guanine. This is guanine. First carbon attaches with guanine and third carbon attaches with OH as a functional group. OH as a functional group, and before nucleotide CH2OH is connected with phosphoric acid. CH2OH is connected with phosphoric acid. This phosphoric acid connected with this OH by the help of phosphodiester bond. Phosphodiester bond. Next, nucleotide. Carbon number 1 attaches with nitrogen base. Thymine attaches with that is adenine by the help of double bond hydrogen bond. Double bond hydrogen bond because of thymine attaches with adenine. As third carbon attaches with CH2OH. CH2OH is freely attached, and here CH2OH is connected with phosphoric acid. Phosphoric acid. This phosphoric acid binds with CH2OH by the help of phosphodiester bond. Next, another last nucleotide. This is first carbon attaches with nitrogen base. Guanine always binds with cytosine. Triple bond cytosine. So first carbon attaches with nitrogen base cytosine and connect with the opposite nucleotide, that is guanine by the triple bond hydrogen bond. And third carbon consists of hydrogen and hydroxyl group. Hydrogen and hydroxyl group. Next, here before nucleotide consists of that is CH2OH as a function, CH2OH as a freely attached group. This CH2OH of fifth carbon connected with phosphoric acid. Connected with phosphoric acid. This phosphoric acid is connected with third carbon of functional group. By the phosphodiester bond releases water molecule. Here CH2OH is again freely attached to fifth carbon. This is CH2OH is freely attached. So this is third prime to fifth prime. So it is anti-parallel. So this is fifth prime to this is third prime. Third prime to fifth prime and fifth prime to third prime. Both are anti-parallel direction. Both are anti-parallel in direction. So pentose sugar connect with nitrogen base, and adenine always binds with thymine, double bond hydrogen bond. Next, cytosine always binds with guanine by the help of triple bond hydrogen bond. Next, thymine always binds with adenine by the double bond hydrogen bond. Next, guanine always binds with cytosine by the help of triple bond hydrogen bond. This is the pentose sugar. These are the phosphoric acids. Next, this is the reverse nucleotides, and this is the pentose sugars, and these are the phosphoric acids, and these are the nitrogen bases. So three things are covered. One is pentose sugar and pentose sugar, 4 pentose sugars. Next phosphoric acids. Next here also 4 pentose sugars, 4 phosphoric acids. Next 4 nitrogen bases connected with 4 opposite nitrogen bases by the triple bond and double bond hydrogen bonds. This is structure, I mean molecular structure of DNA. What about general structure of DNA? Actually DNA is a double helix structure, mainly anti-parallel in direction. Both are anti-parallel in direction. So this is DNA double helix structure. This is about DNA double helix structure, which is mainly contain third prime and opposite helix, I mean another helix, that is a fifth prime. Here fifth prime, their opposite third prime. This is next. Nitrogen bases are attached at middle. So adenine always binds with thymine. Next cytosine binds with guanine. Next thymine binds with adenine. Next guanine binds with cytosine. One sequence. Next adenine, thymine. Next cytosine binds with guanine. Next thymine binds with adenine. Next guanine binds with cytosine. Next adenine binds with thymine. Next what is that? Thymine binds with adenine. Next cytosine binds with guanine. Next guanine binds with that is cytosine. Adenine binds with that is thymine, and here cytosine binds with guanine. Next adenine binds with that is thymine. This is about DNA structure. I mean nucleotides are mainly formed between, nucleotides are joined together in this helix. In this part nucleotides are present. Next, I mean these are the nucleotides are present in this helix, and these nitrogen bases are present in this part. So if we have to this, I mean length between these two strands is 20 angstroms. The length between these two strands, that is 20 angstroms. Next the gap between this 3 to 4 angstroms means adding one nitrogen base to another next step of nitrogen base, that is 3 to 4 angstroms, means they are conjugatives, mainly conjugatives. Next here we are observed two grooves or two deeper like structures. This is one, this is shallow groove, this is deep groove, shallow groove and deep groove. This is shallow groove and this is deep groove. Means shallow groove is a small groove, means so this is shallow groove, and small deep groove. This is small, shallow. Next deep groove. This shallow groove and deep groove mainly consist of 34 angstroms. 34 angstroms, and one turn is equal 360 degrees. 360 degrees is equal to 10 base pairs are present. 10 base pairs are present in one turn. One turn means this is like a clockwise. Mainly it is a clockwise. Clockwise means the helix mainly like a clockwise rotating. This is axis, it present at middle, axis. So this is axis. So this is clockwise, and shallow groove and deep groove consist of 34 angstroms. 34 angstroms means they are one turn. One turn means one rotating helix. Both are anti-parallel each other, both are coiled, but both are not cross each other. They are anti-parallel direction. Both are anti-parallel direction, but both are not even cross. They are not cross each other, only they are anti-parallel in direction. So something is the between two strands, that is 20 angstroms. Next, between the nitrogen bases to another nitrogen base step, that is 3 to 4 angstroms, that is these both are consecutives. Next, small groove is there, that is shallow groove, and deep groove is there, deeper groove. Shallow distance between shallow groove and deeper groove, that is 34 angstroms. This groove mainly is one turn. One turn is nothing but 360 degrees, means that is shallow groove to another shallow groove consists of 360 degrees. In this 360 degrees, 10 base pairs are present. In 360 degrees, 10 base pairs are present, and thickness, distance is 334, that is angstroms. So this is DNA structure. In next class we are discussing about how nucleotides formation, Chargaff rule, base pair rule, rules we are discussing about in next class.